Thank you for coming. My name's Alan. I teach in the School of Pharmacy just across the road. When I'm not doing that, I also do research into cancer. And it seems a bit strange to have what's called a fun day and I'm talking about cancer, but I'm not sure I see the fun part there. But it's what I do. And we were asked, can we do a presentation for the fun day? And I do cancer research, so it's what I can do. I'm sorry. I can't juggle anything like that. What I'm going to try and do today is explain the obstacles that exist to treating cancer. And I'll tell you up front, there's some good news and there's some news that's less welcome. You know, we all probably know someone who's suffering with cancer, or maybe some people in the room are suffering themselves. So there is some bad news as well as some good news here. So I just want to be upfront with you about that right now. There definitely is some good news though. So, what problems are we having treating cancer? The first thing to understand is that cancer is actually a disease of our own bodies. It's unlike, say, a bacterial infection. If you get a bacterial infection, that's a foreign organism. Maybe it looks something like this. It's very different from our own bodies. So if you think about what you want to do with a bacterial infection, maybe you cut yourself and bacteria have got in your wound. To be brutal, we want to kill the bacteria, don't we? Well, when you get cancer, we want to do the same thing. We want to kill off the cancer cells, to be blunt. And the problem is that if you're trying to kill off bacteria, they're so different from us, it's relatively easy to make drugs that kill the bacteria, not us. When we're talking about treating cancer, it's a lot harder because it's a disease of our own bodies. So it's harder to selectively kill the cancer cells and avoid doing damage to our own bodies. And in a way, I could stop the talk right now and say that there's the explanation why we haven't completely cured cancer yet, because that's probably about 50% of the explanation, in my opinion. But there's a bit more to say, at least. So there's the first thing we need to think about. It's a disease of our own bodies. What I want to do is present about 5 slides reminding you of some biology you might have learned at school, or have forgotten, or never learned, I don't know. Very simple biology. It will allow the rest of the talk to make sense. So if you know it already, I apologize. So when cells divide, when cells grow, when a tumor grows, they do so by dividing in 2. So here's one cell, say it's in a tumor. That cell's going to get larger, and it's going to divide into 2 cells. And then those 2 cells are each going to divide. So you've now got 4 cells. So the cells are multiplying. So that's how a tumor gets bigger. The cells divide repeatedly into 2. 2, 4, 8, 16, 32, and so on. That's the first easy lesson. Next one, we need to think a bit about DNA. We need to think about what DNA is and what it does. So I think of DNA as, it's actually quite appropriate we're in a library, because I think of DNA as being like a library. It contains the instructions to make proteins. Proteins are the things in your bodies that actually carry out most of the biological processes. They're the workhorses, if you like. And the DNA contains the instructions how to make those proteins. And it's actually contained in a series of letters, if you like, chemicals that are in the DNA in a certain sequence. There's 4 of them that are possible. We call them A, T, G, and C for short. And they're in a certain sequence. And it's that sequence that contains the information for making the proteins. So you might get a sequence that reads something like ATG, CAC, TT, whatever. That's the code for making a protein. And if you're a cell, you know how to read that code. Now, between the DNA and the proteins, there's something called RNA. And that we're going to come back to at the very end of the talk, but I want to talk about it now to introduce it. RNA is like a messenger. It's a copy of the DNA that carries the message into the cell, telling the cell to go make the protein. And I was thinking, well, what's the best analogy I can come up with for this? And I came up with this idea. Suppose you go to a restaurant and you sit down at the table and you want to order your food. But this is a bit of a strange restaurant. You don't get a menu. They put the entire recipe book in front of you. Jamie Oliver's How to Cook or whatever. And instead of saying, well, I want you to cook me this risotto, what I want to do is I'm going to give you the whole recipe. And the waiter takes the recipe to the kitchen and the kitchen makes it. So the waiter's like the messenger, like the RNA. He's like the RNA. He's taking the information that's coming from the DNA, the cookbook, and the waiter's the messenger taking it into the kitchen to go make the protein. So there's the information. The RNA is the message. It's got that information in it, and then it gets converted into a protein. Does that make sense? Okay, cool.
And is the waiter informed by the RNA?
The waiter is kind of carrying the RNA, so yeah. Now, actually, we might need to kill the lights because I've got some red text. I didn't think about this. So the DNA can mutate. This is where cancer comes from. The DNA can mutate. And when the DNA mutates, the code gets altered. When the code gets altered, the proteins get altered and they don't work properly. They start to get things wrong. And when they do that, instead of controlling the cells in the way they're supposed to, they make a mess of things. And that's what leads to cancer. The uncontrolled processes that occur because the code has mutated. And the processes that get altered are shown in this diagram here. This is I've taken it directly from a paper that's published 24 years ago. And it's one of the most important papers in cancer biology by two guys called Hanahan and Weinberg. And I'll read it out because I can see it's a bit blurry. It lists the key features of cancer cells, the things that go wrong. So this says, it says sustained proliferative signaling. What that means is the cells just keep growing. They divide and they divide and they divide. And they don't need a signal to tell them to do it. They just keep doing it all on their own. And they ignore signals saying stop growing. So they keep growing. Now, cells can also sometimes, and this is going to sound a bit strange, commit suicide. They don't do that, cancer cells. They mutate and ignore signals saying, hey, go kill yourself. And the mutations can also cause the cells to spread around the body, so you get tumors in multiple places. And they can live the cells to live essentially forever. So I have growing in my lab down the road at present cells that were taken out of a patient in 1955, and they're still growing. Okay? And there's no end in sight. They can also, the changes also that take place in the DNA can also cause new blood vessels to be formed because tumors need to get nutrients like anything else. So these are called the hallmarks of cancer. The genetic changes trigger these hallmarks, and that's what allow the cancer to develop. And then the last bit of the tutorial I need to do before we get into what cancer is the idea that DNA in cancer is continually mutating. So here's our DNA. You get a mutation. And normally, our body has a way to fix the mutations. It's got a built-in error checking system. And they're called DNA repair genes. They fix the mutations. So if you get a mutation, the DNA repair genes kick in, and they go and fix those mutations. So that's what that's supposed to signify. Now, mutations can occur in the DNA repair genes, which mean they do a less good job of fixing the mutations. So that allows more mutations to occur, some of which may occur in DNA repair genes. So the DNA repair system is even more compromised. And so we get even more mutations. So we can get a kind of snowball effect. Once the mutations start taking place in the DNA repair genes, we can accumulate more and more mutations. And as a result, cancer cells can be extraordinarily mutated. I'll show you some of that later on. By the way, it's thought that every single cell in our body has around about 10,000 mutations per day. Quite a few. And the reason we exist is the fact these repair genes kick in and fix most of the mutations. Okay, so that is all of the background. Okay, that's all you need to understand the rest of the talk. That's the biology lesson over. Hopefully that was, is that okay? Everything make? Okay, good. So, why haven't we completely cured cancer yet? I put completely in brackets because some people do get cured, okay? Absolutely. But not everyone. And we've been trying to cure cancer for a long, long time. We started using surgery seriously just before the beginning of the 20th century when anesthesia became available. Radiation was developed a little bit later. And then chemotherapy, which is perhaps still the mainstay of modern therapy, was only invented just after World War II. Since then, we've developed something called hormonal therapy. But the more recent therapies, the stuff I've worked on is targeted therapy, and then more recently we've got immunotherapy and RNA vaccines. I'm going to talk about all of these in the talk today. I won't talk about hormonal therapy. If you want to know about that, I can talk to you afterwards. So let's look at chemotherapy first. Chemotherapy has an interesting history. Chemotherapy are actually quite toxic compounds. So to explain the origins of chemotherapy, we need to go back to World War I, when people were fighting in trenches, and the military thought it might be a nice idea to shoot shells at the opposite side with poison gases inside them and poison the troops. And both sides did this until they discovered it wasn't a great plan because the wind could blow the gas back on your own troops. So they stopped doing it. Come World War II, we were more civilized, I suppose, and it decided not to shoot toxic gases at each other. But both sides still stockpiled these toxic gases. They had them ready to use in case the other side did. And in 1943, a ship sailed from the States to Italy with a consignment of these toxic gases. And it anchored in a harbour in a town called Bari. Now, unfortunately, the German Air Force, the Luftwaffe, came over that night and bombed it. And it released the toxic gases into the town of Bari. And, of course, the sailors on the ships were exposed to it as well. And the doctors were completely mystified by what happened to the people who got exposed to these gases. And they found that over the following years, they had a decrease in the number of white blood cells. So the cells in your blood that are part of the immune system and part of fighting infections. The numbers of white blood cells just decreased. Now, a few years later, someone working on cancer realized, well, you can get cancers where you get too many white blood cells. So hang on a minute, let's remember that ship. And here it is. That's the ship in the harbour in 1943 releasing its gases. That gas decreased the number of white blood cells. So if people have got too many white blood cells in some cancers, maybe we can modify those gases and use them to treat cancer. And that's what they did. That's where chemotherapy came from. Which is quite astonishing, really. It's obviously not the same chemicals precisely, but that's where the ideas came from.
Was that truly just chance, the eureka?
Sorry?
Was it truly chance? I mean,
There was one doctor who spotted it and he believed it and he argued it, and everyone didn't believe him. I forget his name for the moment, but ultimately it's proven right. Yeah.
yeah. I've been there.
Well, I'm going to show you the very last slide. There's going to be another surprising feature, moment in history. So, the chemotherapy, the cells divide. Chemotherapy is used to preferentially kill dividing cells. So what happens when cells divide? They have to make copies of their DNA. So here's one cell, it's going to become two. So it's got to make more DNA so it can produce two cells. What most chemotherapy does is it interferes with this process. It interferes with the production of DNA, or it damages the DNA. So the DNA, instead of being copied accurately, it's either damaged or the cells can't make it. Now, when cells try to divide, they have something called a checkpoint. So at this point here, they stop and ask themselves, okay, is my DNA in good shape? Is it okay to go ahead and divide? Because I don't want to divide if I haven't made two copies and everything's okay. And because of the damage that the chemotherapy causes, the DNA isn't in good shape. And the cells at this point realize they're damaged, and actually they commit suicide. And so chemotherapy works by causing DNA damage in the cancer cells, and then the cells commit suicide. So that's what happens in the cancer cells. So when we use the chemotherapy, we can wipe out the cancer cells. The problem with chemotherapy is this. Although most normal cells aren't rapidly dividing, some are. So here are the normal cells. Unfortunately, this cell here is in the process of dividing. Most of them aren't, but this one is. So the chemotherapy has an effect on some of the normal tissue as well. And that's why chemotherapy can have sometimes very nasty side effects, because it's killing some of the dividing normal cells. It works best on the dividing cells, so it kills the cancer cells very effectively, but there's some collateral damage. And in principle, you could kill every cancer cell in the patient's body with chemotherapy, but the amount of damage you do to the patient would be unacceptable. So we're limited in the amount of chemotherapy we can use. So around about 1970, 1980, people started to think, well, can we do better? Can we find more selective drugs that have less disastrous effects on normal tissue? And they developed something called targeted therapy. And targeted therapy is a type of therapy which goes after the genes that are driving the cancer. Do you remember I said the genes can get mutated? Or some of them can actually act as drivers of the cancer. So this is a very complicated diagram which you may not be able to see. It shows signaling pathways inside a cell. So this is the outside of the cell, this is the inside of the cell, that's the surface of the cell. And you probably can't read any of this down here, but this says cell proliferation. So this pathway coming down here is the pathway through which cells are told to go and divide. What can happen in cancer is the genes encoding these proteins can get mutated, so they're switched on all the time. So they're driving this pathway all the time. So the cells are continually proliferating all the time because this pathway is always switched on. So what we figured was, well, hang on a minute, if we can identify these genes that are switched on all the time, the things we call drivers, they're driving the cancer, if we can identify these drivers, surely we can make drugs that inhibit them and stop the cells proliferating. And so that's what happened. We started to make drugs that inhibited these drivers. It's around about 500 of them, we think. So we need to come up with at least 500 drugs. More, actually, but we'll get to that later. But we think there's probably around about 500 genes that can drive a cancer. And this turned out to be reasonably successful. So in 2001, what most people would call the first targeted agent was approved. It's to treat leukemia. It's a drug called imatinib, or sometimes called Gleevec, if you call it by its trade name. And it made the front cover of Time. That's actually my copy of the Time magazine from 2001. And I've still got it at home actually, we were very, very enthusiastic because we thought, hey, this is great. I think in the clinical trials, 31 out of 32 patients were cured of their leukemia. And leukemia, this type of leukemia, is a very difficult one to treat. You couldn't treat it otherwise. It got approved on the basis of just those 31 out of 32 patients being treated effectively. So we thought everything's great, it works. But unfortunately, it's not so simple. So in order to understand why it's not so simple, you need to remember the two things I've told you. That cancer cells grow by dividing, and the DNA in the cancer cells can mutate, okay? And don't forget, we've got these drivers, these genes that are drivers. So can we turn the light back on again? Because I'm going to do a demonstration. So, these are supposed to be cancer cells. So, let's say this is our patient, and the yellow balls... Oh, yeah, good way. No, you're okay. Is that all right? And the yellow balls are going to represent the normal tissue. And then, let's say the blue balls are going to represent the cancer. So let's put the cancer in there. So let's see how chemotherapy works. Well, chemotherapy kills the dividing cells, right? So we just start killing off the blue cells. Because they're the ones, the yellow ones, the normal cells aren't dividing. So fantastic, it's all working. But unfortunately, we got an orange one. And that's one of our normal cells that happen to be dividing. So we've caused some adverse effects, we've caused some nasty side effects for the patient. Okay? And we don't want to keep on doing that, so we can't kill off all the tumour, because if we try to kill off the tumour, we're going to get some of the orange, more of the orange normal cells as well. So that's chemotherapy. Let's look at targeted therapy. So do it slightly differently this time. We'll forget the normal cells. So here's the first tumour cell. And the colour, it's red, that means it's driven by a gene. Let's call it a red driver gene, okay? The cells are driven by a gene, and we're going to call it a red driver gene today. So, okay, it divides. You get one cell, then you get two. 2 become 4. 4 become 8. And 8 become 16. 1, 2, 3, 4, 5, 6, 7. But you remember I said that DNA mutates. So this one's going to mutate, and it's going to be blue. Okay, so now the patient has got a tumour, and they turn up at the doctors, and the doctors think, okay, I'm going to treat this. So they take a biopsy, a sample. Oh, look, they pulled out a red ball. Okay, this cancer's driven by the red gene. We're going to use a drug that kills red cells. Okay, so let's start doing that. Let's start killing off the red cells. Here we go. And it's working. Look, the tumour's shrinking. Fantastic. We've shrunk the tumour. Let's suppose, though, we don't get it all. We left a few behind. So they start growing again. So it starts to grow back. So, of course, the blue cells go divide as well. But the red becomes 4. Then they're going to divide again. So we need another 4 red. And now we're going to have another 4 blue. And then I'm going to divide again. So I need 1, 2, 3, 4, 5, 6, 7, 8. So they divide again. So the red cells are divided, and I need another 4 blue. So now the patient comes back and says, look, my tumour's come back. So the doctor says, well, okay, we used the red drug last time, let's lose it again. So it works. Okay, fantastic. Look, the tumour is shrinking. It looks like it's worked. Tumour's got a lot smaller. Okay, that's fantastic. So the patient goes away. But the cells start growing again. So the red cells divide. And so do the blue ones. So I don't know how many blue ones we want, but it's going to be something like this. And maybe one of the blue ones mutates and becomes a green one as well. Okay, let's have a few more. Let's suppose they divide again. So chuck in a few more blue. We need another green and a few more red. So something like that. It's about right. Haven't got the numbers perfect, but it'll do. Patient comes back and says, my tumour's come back. So the doctor tries his red drug again. And the tumour shrinks a little bit. But not completely. It's what we'd call a partial response. The tumour's got smaller, but these cells haven't got the red driver gene, so they're not affected. So the tumour starts growing again. We get the green, we're going to get some more green cells, we get some more blue cells. And maybe we've got a yellow cell from one of the blue ones this time. Okay, so it's something like that. Patient comes back. The doctor's not going to try the red one because it didn't work last time. But now he takes a sample. Oh, we need to use a blue drug. So we now start treating with the blue drug. And the tumour shrinks. And it's going to shrink an awful lot, which is great. So now the tumour's got a lot smaller. Suppose we do a really good job of it with the blue drug. And we've got down to this. So the patient goes away happy, the tumour's shrunk. If it starts growing again, the blue one doubles, the green one doubles, and the yellow one doubles. And then that happens again. They double all over again. So I don't know how many green balls I need at this point, but it's going to be something like that. there's no point trying the red drug, is there? It ain't going to work. Try the blue drug, which worked last time. But it's not really done a great job, because now this tumour is actually predominantly driven by the green driver. So we started off with this tumour that's sensitive to the red drug. Then it evolved and became sensitive to the blue drug. And now the blue drug's not going to work really, but the green one might. And that's the problem we've got. Cancers evolve. We throw therapy at them and they adapt to that therapy, because the ones who've got some advantage grow back out again. And that's why tumours come back. That's why they become resistant to therapy. Does that make sense? That's how I was going to explain this if I didn't have access to the board. So that's a quick summary of what I said. So it's actually even more complicated than that, because you might have two patients. This patient's got a cancer that's driven by the red gene, and this one's got one driven by the lilac gene, maybe? They need different drugs. They're not both going to respond to the same drug. And this poor chap, he's got one tumour down here that's driven by the red drug, but that tumour spread, and as it spread, it mutated. It's now got this tumour that's driven by the purple gene, sorry. So he needs two separate drugs, actually. And this person, some of his tumour is driven by red genes, and some of it's driven by kind of browny gold genes. So he needs two different drugs as well. And it can get worse than that still. Because if you just look within that one tumour, and the little blue things are supposed to represent the cells in the tumour, within each cell there might be multiple drivers. You know, I said there's 500? We think in average in cancers there's up to 5 driver genes. Maybe 6, maybe 7. Leukemia, blood cancers, probably only 1 or 2. But in solid tumours, there's probably several driver genes. So we need more than one drug. And every time we throw a drug at the tumour, it evolves, as I showed you with the diagrams there. So they evolve over time. So, can we kill the lights again? So instead of showing you with balls, I'll show you something for real, what it really looks like. So here's that signalling pathway, and this protein signal at the top is called the EGF receptor. It can send signals down this pathway. Here's what it really looks like. In green is that receptor. It's a structure of the protein. We don't need to go into all the detail today. But the blue thing is the drug bound to that protein, turning it off. Okay? And what can happen is the thing I've coloured in there, in yellow, can mutate. And when it mutates, it gets bigger, and there's not enough space for the blue drug to bind. So patients turn up at the clinic, they respond to this drug, which is called gefetinib. Then this yellow bit mutates, there's no room for this to bind anymore. In fact, it looks a bit like that now. And this drug doesn't work anymore. But up the road at AstraZeneca, they developed another drug called osimertinib that actually binds even though the mutation's present. So the patients start off on this drug, it works, a mutation occurs so it doesn't bind anymore, and then you switch them over to this therapy, and then probably another mutation takes place and you have to switch them over to a third therapy. So one single drug doesn't get rid of the tumour. Those are real structures of proteins and real structures of drugs. So that's targeted therapy. I thought I'd also talk about the immune system, because it has been, in some patients, extraordinarily successful. And if you think about what the immune system does, is it attacks foreign objects. Okay? It identifies something as foreign and then attacks it. Now, if you remember the very first slide, I said cancer is a disease of our own bodies. It's not foreign. Why on earth would the immune system attack it? And I'll be honest with you, when I was early on in my scientific training, I thought this whole immunotherapy is nonsense because cancer's disease of your own body. Why the heck is the immune system ever going to attack it? I was wrong, which is okay. This is good. Remember the DNA mutates? Okay. Well, when the DNA mutates, the proteins mutate, and now they start to look foreign. because they're no longer identical to our normal proteins. They're looking like they're foreign. So the immune system can hook onto them and attack them. And they start to do it. However, there's a problem. And that problem is that cancer is a chronic disease. It goes on for a long period of time. And the immune system, if you like, gives up. It gets, I don't want to say it gets bored, but it's something like that, okay? It takes a break, let's put it like that, and stops attacking the cancer. What immunotherapy does is to reawaken the immune system. So this is a diagram I stole from someone at the Crick Institute. This is supposed to be a cancer cell. This is supposed to be a cell in the immune system attacking the cancer cell. And the cancer cells are sneaky because they can make a protein, which is this orange thing here, that binds to a protein on the surface of the cell in the immune system and says, hey, don't attack me. Leave me alone. Go back to sleep. The best analogy I can draw for you, if you've ever seen the movie Star Wars, there's a bit in that, that's the very first Star Wars movie, it's probably the only one I watch really, but where Obi-Wan Kenobi says to some Imperial guards, these are not the droids you're looking for, and they go straight past. This is what's going on here. The tumour's saying to the immune system, I'm not the cells you're looking for. And it sends the immune system to sleep, if you like. What we do with immunotherapy is we switch the immune system back on, and the immunotherapy, this green thing here, is turning off this signal which is sending the immune system to sleep. And so we reactivate the immune system and it attacks the cancer cells again. And that's called immune checkpoint therapy. And it works very well, but only in some patients. Now there's another type of immune therapy we use called CAR T therapy, which is probably another 10-minute 10-minute explanation on its own, but basically the idea is we can engineer cells in the immune system. That works slightly differently, so I won't go into that today. If you want to know about it, I can explain it later. But the thing that's used most commonly today is this type of immune checkpoint therapy where we're reawakening the immune system. And in some patients, it works really, really, really well. But in some patients, it doesn't work at all. And this is one of the probable explanations why. And this is I pulled this out of a scientific paper. It's from a journal called Nature, which is one of the best scientific papers journals there is. What it does is it lists down the left-hand side here different types of cancer. And going across this top bit here is how many mutations there are every million bases. So if you remember, I said the DNA's got a sequence of letters in it. So this is how many mutations when you look at every million letters. So that would be one mutation every letter, every million, and that would be, well, let's say this one, that's about 1,000. Each black dot is one person, or one person's tumour. So this person, let's go with this one, this person had around about 1,000 mutations every million bases. That's an awful lot. If you look down here, they've got around about 10,000-fold less mutations. And all the others are somewhere in between. And even if you look at just one type of cancer, this is melanoma, there's about 1,000-fold variation in the number of mutations you get. So one of the reasons immunotherapy might not work is that some cancers are not as highly mutated as others. If you've got a highly mutated one, then the immune system's got something to hook onto. If you haven't, then it can't. Last type of therapy I thought I'd tell you about today are the RNA-based vaccines. And the only reason I'm telling you about it is I heard about it on Radio 4 about a month ago, if I'm honest with you. And this is something that's just gone into clinical trials. People are volunteering to receive this as we speak, I guess. And it's driven by technology that you have probably heard about during the COVID pandemic. So you might remember from the COVID pandemic, there was something called an RNA vaccine. Let me explain how vaccines work. Normally, here's our DNA, here's our RNA, here's our protein. Normally, when you want to use a vaccine, you make the protein in a lab somewhere. So if I want to immunize someone against measles, I go and make some measles proteins in the lab, and then we go and jab someone with it, and their immune system recognises it's foreign, and they're ready for the next infection. So you're using the protein to educate the immune system. That works fine, but making proteins is technically difficult. It just takes time and it's hard. We can do it, but it's just complicated. Making RNA is actually quite easy. I could probably go back to my office, go and type something into my computer and have some by Monday morning. Well, maybe Tuesday, it's a weekend. So some of the COVID vaccines were developed very quickly because all we needed to do was look at the sequence of the COVID virus, look at the mRNA, or the RNA, sorry, and we could design vaccines quite quickly and make them quickly. That's why we were able to come up with vaccines quickly against COVID, because we've changed the technology. So these are called RNA vaccines because you inject the RNA into a patient and their cells then make the protein, and then the protein switches on the immune system. It wakens up the immune system. So we can adapt that to cancer, and that's what's going on at present. So you take a tumour from a patient, you go and sequence the DNA, you make some RNA, so the sequence corresponds to the mutated regions, you make some RNA, inject that into the patient, and now they will have an immune response against that mutated protein and will hopefully then attack the cancer. That's the idea. And the only reason this is working, or the only reason this has been tested, is more accurate, is that we've got this new technology where we can make the RNA vaccine very quickly. We couldn't do it by making the protein, because if a patient has cancer, you take the tumour out of them, you sequence it, and you want to go make the protein, that's going to take weeks, months. This can happen really quickly. So it's potentially going to be game-changing, but my guess is that it will only work for some patients, the ones who've got highly mutated tumours. So that's all I'm going to say. I'm going to end up with 2 more slides. This one's a lot of text. It's a summary describing everything I've just said. So cancer is a disease of our own bodies. That makes it difficult to treat. It's not just one disease, because there are multiple genes that can cause it. And as I showed you with the demonstration, it evolves over time. It's a moving target. One thing I haven't touched on is it's expensive to make new drugs. Anyone want to take a guess how much it costs to make a new drug? 2 billion. I'm off the top of my head, I think the budget for the NHS is around about 160 billion a year. So 1/80th of the budget of the entire NHS for the entire country is how much it costs to make one new drug. That's why drugs cost a lot. Oh, and the other thing, no one ever mentions this, I don't know why, maybe because we're embarrassed. 90% of drugs that go into patients to be tested actually don't work. And only 10% succeed. So you're actually also paying for the failures. So drugs are expensive. Then we need to have a healthcare system to support cancer treatment. Early detection is really important, because surgery is probably one of the best ways to treat cancer if you can detect it early enough. If you can cut it out before it develops, before it spreads, that's probably one of the best ways to treat it. And we have an ageing population. So we have to adapt our healthcare system to summarize that, to deal with that. But we're making progress. Survival rates have doubled over the past 40 years, so that's good news. And I happen to work in ovarian cancer, so this is the example I'll quote to close. This drug called olaparib allows 65% of ovarian cancer patients to live 7 years or more, which is a tremendous step forwards. Last slide, this one. It's a plug for my own website. It's not about cancer, it's about science in general. If you're interested, please go take a look. If you want, there's some cards. You're welcome to take one with you. It's got the address on it. If you want to understand scientific principles, please go take a look.
Just go back to that previous slide. Sorry, it's with defective HR.
Defective HR. So,
Defective HR, what does HR stand for?
homologous recombination.
Oh, right.
So, but I can explain it easily. Do you remember when I showed you about 20 slides back that you can get mutations in the DNA repair genes. That's what HR is. It's one of the DNA repair genes. And it's a beautiful piece of science which makes sense, but when I teach this to the undergraduates, it takes about half an hour for them to get it. This drug only works if you've got a HR system, a DNA repair system that's not working. And it's hard to understand that, I know.
It just caught my eye and I thought, oh, well, perhaps we'll know that, so thank you.
Yeah, sure. Happy to answer any other questions.
Are there any other questions from the room? We don't seem to have any online at the moment, Alan.
That's okay.
If any of our online guests would like to ask a question, they're more than welcome to as well. Really interesting talk. I'm going to ask quite a difficult question, I think, and I don't know whether you'll have an answer for me, Alan, on this one. Is there any kind of research behind why 1 in 2 people are likely to get affected by cancer? Because I think that's a statistic we hear a lot as the general public with no knowledge, but I've never really known where that statistic comes from as such.
Well, the statistic comes from observation, but it's looking at how many people get it. There's 2 components to cancer. At least you can think about it like this, I think. What you inherit and what you get exposed to in your lifetime. So you can inherit genes that increase the chance of you getting cancer. So there's a type of gene called a tumor suppressor. And what those genes do is they prevent cancer forming. Now, you might not know this, but for every gene we've got 2 copies. Okay? Because you get one from your mother and one from your father. Now, the tumor suppressors stop the cancer forming. So here's my 2 copies of my gene. Fantastic. 2 tumor suppressor genes, both are going to work to stop getting a cancer. If I inherit 1 defective tumor suppressor gene, I've only got 1 now. And if that one happens to get mutated, I've now got none. So I only need 1 to get mutated compared to a person who's got 2 before I lose my tumor suppressor genes. So you can inherit defective tumor suppressor genes, and that increases the risk of you getting cancer. The most famous example of this is probably Angelina Jolie, who had a mutation actually in the HR pathway. And she knew, therefore, she was likely to get, or at risk of developing breast or ovarian cancer. And so she had a double mastectomy to reduce that risk. So losing your tumor suppressor genes doesn't cause cancer, but it prevents them from stopping cancer.
Was it, sorry, the statistics weren't just always high as 1 in 2. I mean, have they been that high?
It's because we're getting older.
Yeah, but I mean, 10 years ago, was it 1 in 2?
I think it was 1 in 3 was the estimate. It's because we're an aging population, and cancer is a disease of old age because you accumulate mutations over time, okay? We just, I said we get 10,000 mutations per cell per day. That just builds up. And over time, we get more and more and more mutations. So it's a disease of old age because we accumulated more mutations.
And isn't it the case that many people will die with cancer, but not of cancer, but it's more of the have been diagnosed, you've got it, but that's not actually what gets you in there. Maybe some might be heart or whatever else goes as well. Yeah. But we're just aware that...
Yeah. Yes, you can die with cancer but die of something else instead. The classic example, for the men in the room, I'm sorry, but most men aged, I think it's 80, who have got beginnings of prostate cancer. So,
Can I ask something?
Even if it's not overt cancer, yeah, sure.
So my mum unfortunately succumbed to cancer, but in very, very old age. So at 85 she was diagnosed with breast cancer, which was treated by this magic pill that reduced the tumour to its roots and provided she took the daily pill. But she ultimately died from ovarian cancer, which spread to her bowel. And I suppose my question is there a direct link between breast cancer and ovarian? The fact that she had breast cancer at 85, which was successfully treated in terms of reducing, and then 5 years later eventually developed symptoms that she was very unwell with something, and they discovered she had, probably quite a large ovarian tumour.
Yeah.
I'm pretty sure it was the ovarian tumour that then spread to the bowel. We think it's that way around.
Yeah.
Is there a link, or have you any idea if there is a link between the breast cancer that she had at 85 and developing ovarian cancer?
So I think there's two possibilities. One is it was two independent events, so two completely separate cancers. That's certainly one possibility.
Yeah.
There is another possibility. The BRCA gene I mentioned that was mutated in Angelina Jolie, if you get mutations in that, that can increase the risk of both breast and ovarian cancer. That's a possibility.
Right.
There may, do I know any other genes that drive both? Not off the top of my head, but there's
So what
certainly.
I'm hoping is that it's not the second one you mentioned, because the BRCA gene, because that I could have that.
Yeah.
Right.
Yeah. You can, but you can, I'll be with you a second, you can be tested and you don't necessarily have it because you might not have inherited it.
Yes, but you can be tested to see if you have
Yeah.
that gene.
Yeah.
And 85 is quite old, isn't it? I mean,
Yeah.
if the BRCA gene's going to get you, it's going to probably get you at an earlier stage, isn't it?
I think that 85 is quite old, you're quite right, yeah, it's possible. Yeah.
My apologies for sneaking in
It's
late.
all right.
I've been to another event that clashed with this, and
It's
I
okay.
realise I've missed what I think would be a fascinating lecture. I just having caught the last part of it. Am I able to see this online? Is this been saved somewhere? You probably made an announcement about that, but obviously I've missed it.
Yeah, we do. Yeah, we have recordings of the live
So
stream.
I'll be able to go and see it from scratch somewhere online.
Yeah.
Thank you so much.
Oh, pleasure.
Thank you.
You said something about heart of cancer, the symptoms of cancer or catching cancer could be hereditary. Right, so if somebody's had cancer, the chances of their children becoming cancer victims has increased.
It depends. So it depends where, we talked about the idea that cancer is driven by mutations. It depends where the mutations occur, okay? So suppose I get a mutation in a lung cell, okay? My lungs aren't involved in reproduction. I'm never going to pass that on to my children. It's only if you have mutations in the cells, we call it the germ line, basically the cells that give rise to sperm and eggs. It's only if you've got mutations in those that you're going to pass it on. So we distinguish two types of cancer. What we call sporadic cancer, that means it's anywhere in the body, and hereditary cancer, where it is something you actually passed on. And for that, it's got to be in those germ cells, the sperm and the eggs, to be passed on.
Great, no, thank you so much, Alan. And just to say as well, we have had a comment online to say how much of a great presentation that's been. So thank you so much for this talk this afternoon. It's been absolutely fantastic.